All right, today we're going to pull back the curtain on a piece of technology that's, well, it's a total game changer in modern medicine. We're talking everything from fighting cancer to managing viruses, so let's jump right in.
I mean, think about this for a second. How in the world do you take a tiny drop of anything, really, that has millions, maybe even billions of cells in it, and count them one by one at lightning speed? It sounds completely impossible, doesn't it? Okay, before we get into the nitty-gritty science, let's use an analogy. Picture one of those super fast high-tech candy sorters. You know, the kind that can whip through thousands of little candies every single second, checking their color, their size, and then zipping them off into the right bins. Got that picture? Okay, now just imagine those candies are as small as a single human cell. And right there, you've basically got the core idea behind the tech we're demystifying today: flow cytometry. It sounds a little intimidating, I know. So what does that name actually tell us? Well, if you break it down, it's actually pretty straightforward. Cyto means cell, and metry means to measure, so it's cell measuring. The flow part? That just means we're measuring the cells while they're flowing in a liquid. And the magic happens when we shine a laser at them, one by one as they stream past. This lets us learn an incredible amount about every single cell, thousands of times a second. It's wild.
Okay, so how does the machine actually pull this off? Let's do this. Let's follow one single cell on its journey through the guts of a flow cytometer. It's kind of like a microscopic roller coaster ride. So first things first, you need the cells, and they can come from anywhere, really. A piece of tissue, a blood sample, even cells grown in a dish. The key is that scientists have to prepare them in a special way, creating what they call a single-cell suspension. All that means is getting the cells to float around freely in a liquid, not all stuck together in a clump. And this is where the first bit of pure genius comes in. The liquid with our cells gets injected into another, faster-moving stream of fluid. It's called sheath fluid. And because of some really cool physics, a principle called hydrodynamic focusing, this process funnels all those randomly floating cells into a perfect single file line. Seriously, think of cars merging from a 5-lane highway down to a single-lane tunnel. It's exactly like that, but for cells. So let's recap that journey real quick. Step 1, the sample goes in. Step 2, hydrodynamic focusing gets all the cells in a nice, neat line. Step 3, bam, for just a split second, each cell zips through a laser beam. And step 4, as it does, it scatters that laser light in all directions. And super-sensitive detectors grab those signals. And remember, this whole dance happens for every single cell, thousands and thousands of times every second. Okay, shooting lasers at cells is cool, no doubt, but it's just a bunch of flashy lights unless we can actually make sense of it all. So the real question is, what are these signals telling us? How do we translate that scattered light into useful information? It really boils down to 2 key measurements. The first is called forward scatter, or FSC. The easiest way to think about this is like the cell's shadow. The bigger the cell, the bigger the shadow it casts as it goes through the laser. So FSC tells us about the cell's size. Simple enough, right? Then there's side scatter, or SSC. This is the light that bounces off all the little bits and pieces inside the cell, the granules, the nucleus, all that stuff. So a cell with a lot of internal stuff will scatter more light to the side. SSC tells us about the cell's internal complexity. And check this out. This is what you get when you plot those 2 things against each other for thousands of cells. Along the bottom, you've got size, and up the side, you've got complexity. And look what happens. The different types of cells just sort themselves out into these distinct little clouds. You can clearly see these guys down here, the smaller, simpler lymphocytes, which are a huge part of our immune system. And then way up here, you've got the granulocytes. They're bigger and way more complex inside. It's like a fingerprint for different cell types. Okay, this is amazing, but you know, a real-world sample from blood or tissue is messy, it's got all these different cells, plus random bits of junk floating around. So what if a doctor only cares about one of these groups, like just the monocytes right here? How do you zoom in on just them and ignore everything else? Well, they use this incredibly clever digital trick called gating. It's honestly as simple as it sounds. You literally just draw a shape like a digital lasso or a window around the cluster of dots you care about on the screen. Then you tell the computer, hey, see this shape? Only show me the data for the cells inside this gate. Everything else, just ignore it. And this really shows you the power of it. On the left, you've got the total mess. all the data points from everything in the sample. But on the right, you can see someone has drawn these gates, these digital windows around the lymphocytes and the monocytes. Just like that, they've digitally purified their sample. Now, any other questions they ask, any other analysis they do can focus only on those specific cells. It's a ridiculously powerful way to cut through the noise. So, okay, we can line up cells, blast them with lasers, and sort through the data. It's an incredible piece of engineering, for sure. But so what? Why does any of this matter outside of a research lab? Well, this is the part where flow cytometry goes from being a really cool tool to an absolutely essential, life-saving diagnostic powerhouse. Because it gets even better. On top of measuring size and complexity, scientists can add another layer. They can use these special fluorescent tags. Think of them like tiny glowing beacons. They attach these beacons to antibodies, which are like little biological missiles programmed to seek out and stick to only one specific type of protein on a cell's surface. So now, when that cell zips through the laser, not only does it scatter light, but that little tag lights up and glows a specific color. This gives us an incredibly precise piece of information, the cell's exact identity.
And that capability is what makes this a true game changer in medicine. The speed and precision are just unmatched. We're talking about things like pinpointing the exact type of leukemia or lymphoma a patient has, which is absolutely critical for getting them the right treatment right away. For patients with HIV, this is how doctors track their immune system, by counting a very specific type of cell called a CD4 T cell. It's used to spot rare genetic diseases, and even to check if a stem cell transplant is working properly. The applications are just immense. You know, one of our sources really hit the nail on the head. They called it a powerful analytical tool that rapidly identifies and characterizes cells. And that's exactly what it is. Using just light, scattered light and glowing fluorescent light, we get this unbelievably detailed snapshot of the tiny building blocks that make us who we are.
And that really brings us to the big picture here. Today's machines can measure dozens of different things on a single cell, all at once, at speeds that would have sounded like pure science fiction just a generation ago. It really makes you stop and wonder, as this technology gets even more powerful, even more sensitive, what other secrets are our cells hiding, just waiting for us to unlock?